Alright, and welcome to osmotic pressure. So, why do we need to learn osmotic pressure? Well, we try to take advantage of this when we try to control bacteria. We either dry them out, and if they're dried out, where's that water moving in from out of the cell? We try to make them take in chemicals that they might not normally take in. All right, so how do we get to do that, and how are we going to kind of trick cells, right? Okay, and so you might have heard of concentration gradients, or going from a pressure of high pressure to a low pressure, right? Or heat rises, okay? It's all kind of like the same kind of theory. Like heat rises is definitely a different thing, but just those are parts of things that we just kind of take for granted. And we've all definitely heard of osmosis, whether or not we fully understand it or not. Um, it's definitely something that's been in the social environment. Okay, so with osmotic pressure comes some things that seem kind of mundane. Maybe you've seen them before, maybe you haven't seen them before. If you've taken A&P, you've definitely seen them before. But they're definitely worth reviewing, and they're definitely worth remembering pretty much for forever because of how the relationships between the bacteria and the humans are mostly taken advantage through this type of example of osmotic pressure. Okay, so what is osmotic pressure? Okay, in most prokaryotes, and remember we're focusing a lot on prokaryotes, eukaryotes can regulate this function a lot better. Red blood cells being an exception. All right, so morphology is maintained by the cell wall in prokaryotes, and this is a structure found in most of the all of the prokaryotes, but not the eukaryotes. Okay, all of the bacteria rather. Cell membrane protects the cell from changes in osmotic pressure, okay, but only so much, okay. And so osmotic pressure occurs because there's differences in concentration of solutes on opposing sides of the semipermeable membrane. What is our semipermeable membrane in a cell? Well, it's that phospholipid bilayer, okay. And so I mentioned water several times, okay, and water can pass through the semipermeable membrane most of the time, okay, because water is just two hydrogens and an oxygen, okay, so it can fit through most things. But solutes, okay, cannot get through some, okay. This is either through a size difference or a selective cell difference, okay. So when the concentration of solutes is greater on one side of the membrane, water will actually try to go out to that and try to make both sides equal. Okay, so how do we think about that? Well, if you're in salt water, okay, and you put in a balloon of fresh water, all the salt is on the outside. So all that water inside wants to equal the salt on the outside, and so it'll rush out to try and equalize with that. Okay. Conversely, if you have salt water um, in the balloon, water will rush in and try to equalize that. Okay, so this is called osmosis. Okay, and if it wasn't water, that is called diffusion. Okay, so osmosis is just diffusion with water, but you can have diffusion without water. Okay, and so what do we mean by rush in and rush out? Okay, so these are actually called something. So um, if you put a cell into a normal solution that it wants to be in, okay, that would be called an isotonic solution. Okay, what goes in, comes out. Okay, so isotonic just means that it's isolated. Okay, isolated, isotonic solution. It's just in isolation, it's going to be fine. Salts go in, water moves in and out. Okay, in a hypertonic solution, okay, now we're talking about the solutions, not the cells. So in a hypertonic, there's a lot of solutes, okay? And how do we think about that? If a kid is hyper, they're hyped up on sugar, right? They're hyped up on a lot of sugar, okay? So if you're hypertonic solution, you have a lot of solutes. So it could be sugar, could be salt, and pick your poison. All that solute out there, that water inside will move out to try and neutralize them, okay? And in a hypotonic solution, okay, the solutes are not in the water, and that water wants to rush in to that cell to fix it, okay? And how I remember hypotonic solutions is it's going to make the cell bubble up, okay, into a big O. Okay, so hypotonic solution makes that cell go big, okay? And a hypertonic solution, when all that water rushes out, it's going to kind of shrivel it up and cause it to crenate, or that's called crenation, okay? And in a bacterial cell or a plant cell, one with a cell membrane and a cell wall, that cell membrane is actually attached to the cell wall, and so when it starts to dry up, it's called plasmolysis, okay? You can call it plasmolysis if it helps, but basically it's saying cutting or destroying the cell membrane or the plasma membrane, okay? So plasmolysis or plasmolysis is what actually happens when the cell starts to shrivel in a bacterial cell. And in that hypotonic solution with the cell wall, instead of bursting up into a big O, like we memorized it, that big O is going to be stopped, and it's going to kind of like take a big old bursting feature kind of a shape. Alright, so I hope this explained osmotic pressure enough. It's worth reviewing isotonic, hypertonic, and hypotonic again, and go over this video again. Like I said, we try to dry things out. We try and trick things, bacteria and fungus, based on these kind of tonicity changes and osmotic pressures. Alright, so now we'll get into our last two lectures, and they might be more than one video apiece based on what they are. Alright, so keep watching and good luck.